Welcome to the Week of Evolution webinar series. We, as both University Science Club Trio of Evolution Committee, collaborated with many scientists who are experts in their fields to present an evolution theme series to you. We wanted to use the opportunities of the online platform to meet with the professors all around the world.
Our mission was always to spread accurate scientific information and create platforms where science can be freely discussed and told. So when the conditions present in our country are taken into account, it is even more important and crucial to do so.
Therefore, we would like to restate the necessity for free academy, as we still protest and stand against the appointed rectors and their undue sanctions.
Both university members have been standing against this anti-democratic situation for more than a year.
Aside from that, this year we will have 9 seminars between 21st of January and 7th of February. And you can also follow our social media accounts to be posted about the calendar.
Today, we will welcome Professor Andrew Berry and Professor Naomi Pierce from Harvard University. After they are finished with their talks, we will have a short Q&A session. You can ask the questions through the chat and our team will edit and direct them to our professors. Welcome again, Professor Berry.
Hey.
One, it's incredibly nice to be back again, and I want to say a huge thank you to the organizing team for putting this all together. Really appreciate it. And I do want to lend our voices to what you just said, Ece, which is academic freedom is absolutely critical. Academic freedom is not just academic freedom, it's freedom. Freedom from overrule, from totalitarianism, from, fascism. And I really think that the world needs to know more about what the faculty and students at Pozitif University are doing to stand up for just that. So that's one. Please understand we support you in everything you do in that regard. The same thing is maybe a bit closer to home for me in particular, is the work that the Tree of Life organization in Turkey and elsewhere does in general in supporting information over misinformation. And this is what we're here to do in essence, especially in a country like Turkey, and especially I speak as somebody from England, in a country like the United States, where creationism is a powerful and effective lobby and is, misinformation. So yes, congratulations, and please continue with your wonderful work in this regard. The final thing I have to say, by the way, about being online in Turkey is it's incredibly frustrating. I come regularly to Sabancı University to teach, usually twice a year, but of course COVID has prevented that. So my experience of Turkey is this, sitting in my office at home in Cambridge, Massachusetts, and I don't get to eat any patlıcan. I don't get to eat any, I don't get to drink any raka. It's very disappointing, but I look forward to returning soon. Anyway, so thank you for the invitation. It's an honor to be here. Yeah, as you know, we're going to give 2 separate presentations. I'm going to start. Naomi will come second. So what I'm going to do straight away is dive in and share my screen. Okay. As I say, we've got 2 slightly different topics, well, very different topics today. The first one, and by the way, the slides have been translated by Ece Bilgin, for which I'm very grateful. What I've decided to do is just leave everything in Turkish. I'm not going to put up the English as well on the slide. The reason is I'm speaking in English, people. You should hopefully manage to get the English message from me. And the Turkish message, needless to say, will come from the slides. So 2 topics. I'm talking about positive selection, detecting molecular adaptation, which is, I'm about to argue, the heart and soul of Darwinism, of the evolutionary process. And then secondly, Naomi's going to talk about her beautiful work on an extraordinary phenomenon, on the fact that plants, boring plants, can actually smell things. They can respond to odors. I just think it's stunning, and it's a sort of relatively new evolutionary discovery, this. But anyway, it's me to kick off with. So yes, I'm going to talk about detecting natural selection, finding DNA, if you like, in Darwin, if you like, in the DNA. And this is just a brief outline of what I'm talking about. As I say, this will be a short talk. I'll be talking about the difference between negative and positive selection, but negative selection, as I'm about to say, is really boring. So we're going to then talk about positive selection and how we can distinguish positive selection from other forces that change allele frequencies in genomes. But let's start with natural selection writ large. What does natural selection mainly do? Well, natural selection mainly keeps things the same. If you've got a complex entity like you, a Turkish undergraduate, tons of moving parts. Just think of it from that moment of conception, when you were a single cell, and then you have this 2 cells, 4 cells, 8 cells, 16 cells. You have this extraordinary process that transforms that one cell to you. I think the estimate is 50 trillion coordinated cells. If you make a random change to that kind of system, you're going to mess it up. Okay, and just to make the point, and I'm trying to do this in Turkish style, here is your Uncle Mustafa's car. It's from what, the 1980s, perhaps? It's an incredibly simple automobile. Much more simple, obviously, than any living organism. But now just imagine you open the hood and you make a random change. You put in something or you pull out something. What's going to happen? You're going to damage the car. You're going to destroy the car. You're going to make Uncle Mustafa very angry, actually. So that's what mutations almost always do. If they land in a functionally important area, they're going to prevent function so that that individual either dies or fails to thrive and is eliminated by natural selection. So that individual is eliminated, which means the mutation is eliminated. So that's what most of natural selection does. It's negative natural selection, keeping things the same. But that's boring. That's incredibly boring. That's not what evolution is about. It's not about keeping things the same. Evolution, the excitement about evolution, what kept Darwin awake at night was adaptation, how new innovations arise in evolution. So look, simple example, the giraffe. What a ridiculous organism. But think about it, it's an extraordinary idea to grow this super long neck so that you are more effective at browsing on the ever higher vegetation at the top of the trees. Now, so there was no negative selection that produced this. There were mutations in the giraffe genome back in history or prehistory, deep in the giraffe past, that caused some individuals to have slightly longer necks. And those mutations gave those individuals an advantage because they got to eat more vegetation than other individuals. And so those individuals, this is natural selection, left more offspring and so on and so forth. That's positive selection, right? That's new mutations which start rare and become increasingly common because they confer upon their carriers an advantage. So that's what I'm talking about today. How can we detect the action of positive selection in genomes? Okay. Now, the first thing to say, well, you might say that's rather simple. Let's consider that negative selection just keeps the things the same. So if we compare two genomes and we find differences between the two genomes, then maybe those differences come to you courtesy positive selection. Okay, so if we compare two different species, for example, spot the difference for their genomes, then maybe the differences are attributable to positive natural selection. So here's that exercise. Famously, if you compare the human genome to the chimpanzee genome, both obviously now fully sequenced, but we knew this actually before they were fully sequenced, it's about 1% genetic difference. And by the way, obviously the big question is, who do we use as the ideal human being? And I've opted for Miley Cyrus. I hope that's okay with you. The sort of perfect exemplar of Homo sapiens. 1% difference. Now, where does that 1% come from? Is it all positive selection? The answer is no. Now, this is a rather busy slide, I appreciate, but it's an important set of ideas that I'm showcasing here. Here we have an ancestral species, which just a tiny bit of its sequence is ATG. Then one of the descendant species is also ATG. No, nothing's changed. But in another descendant species, we've got AGG. So in other words, there's been a mutation somewhere on the right-hand lineage. The T has changed, the middle T has changed to a G. But it's not just a mutation. What's happened is that mutation has then increased in frequency such that it's 100%. Okay? So, and that's obviously going to take time. All right? So you have the original mutation and then what we call a fixation process. Now, this is the critical thing. There are two ways in which that fixation can occur. It can either be driven by positive selection or, boringly, by what we call genetic drift. Genetic drift is just, it's essentially stochastic sampling error from generation to generation to generation to generation. So these are mutations which aren't negatively selected because they're not deleterious, but they're not positively selected because they're not advantageous either. These are what we call neutral mutations, and their fate is governed by genetic drift. So this is perfectly possible that that green G in this figure is a neutral mutation which has drifted by chance to fixation to 100% frequency in the descendant population. The alternative, of course, is that positive selection played a role. And that's a much more deterministic process. You can see, and it's a much faster process because it's not wandering around all over the place stochastically as drift does. This is directed evolution. So that's the difference. And that's what we're interested in. We've got to distinguish between neutral genetic drift fixations and positively selected fixations. And one great clue, as you can see in the little plots we've got going there, is one is slow and wandering and inefficient, takes a long time. The other is relatively swift and efficient. So I'm going to give you 3 case studies about how this process, how we can disentangle drift from direct positive selection. And the first example is a really cool one. And that is what we call convergent evolution. Convergent evolution is simple. It's when you've got 2 independent instances of the same thing evolving because it's beneficial. Famous example, milk. So, as you probably know, humans aren't, if you like, prehistorically adapted to the consumption of dairy. Dairy has a lot of lactose. It's the main carbohydrate in milk. And we have an enzyme called lactase, which breaks it down. Now, normally, you only encounter milk. When I say normally, I'm talking about before the domestication of cattle. You only encounter milk and lactose as a baby, right? For 2 years or however long you are being nurtured by your mother. And then after 2, you don't drink milk anymore, right? So what does that mean? That means the system has evolved to produce the enzyme lactase while you're consuming lactose. And then at the age of 2, when you stop consuming lactose, the lactase gene switches off. That's cool. That works. But obviously, that's no longer what we want because about, well, starting 10,000 years ago, we domesticated cattle. And so now we have milk and lactose every day of our life, if we want it, not just for the first 2 years. By the way, we think there were multiple domestications of cattle starting, as I say, about 10,000 years ago. But you're familiar with lactose intolerance. Lactose intolerance means you basically have the original system, which switches off the ability to digest lactose at the age of 2. And that means you have gastrointestinal problems associated with the consumption of dairy. Now, what we see, and this is incredibly recent because it's all after 10,000 years ago, when turning the lactase gene off at 2 ceased to be a good idea. Before 10,000 years ago, turning it off at 2 was a good idea. Now, with domesticated cattle, it's a bad idea. So what's happened is we've had the evolution, positive selected evolution, of a change in the lactase switch such that it stays on after the age of 2. But the reason we can be sure this is adaptive, this is super cool, is this has happened in at least 2 of the populations which have independently derived dairy and having lactose permanently available. So in European and African populations, so these are 2 separate domestications of cattle, 2 separate evolutions of what we call lactase persistence, i.e. keeping it turned on. So, and how do we know they're separate? Well, here's the lactase gene, and the gene is indistinguishable. It's the same protein for somebody who's lactose intolerant and somebody who's lactose tolerant. The evolution has occurred in the control region, which I've put here in orange in this diagram. And the key part of this diagram, the control region, is the magenta region, which encodes the switch off at 2 switch in lactase. Now, so let's zoom in and look closely here. If we look in an African population which is lactase persistent, we find a mutation which changes the magenta section, so it stays on. So lactase stays on. Okay, so this, if you like, is disabled the magenta section of the control region. That's the African one. Now let's look at a European population. The European population does the same thing. It produces lactase throughout life. So in other words, lactase is kept switched on, but it's a different mutation. That's the cool thing. It's a different mutation. So what does that mean? These are 2 independent events. The fact they both occurred more or less simultaneously and in an understandable situation, so we know what the selection pressure was, tells us absolutely 110% that this is positive selection in action. Fine. That's one. Convergent evolution is kind of a special case. Let's look at a more general instance of how we can detect positive selection. What I'm going to talk about now is what we call a selective sweep. I'm going to show you a simple animation just to show you the impact of natural selection on a particular mutation, the impact it has on the flanking variation on that part of the chromosome in that region of the genome. So let's go to a cartoon. So here we have a set of chromosomes in the population. We've got 3 variable positions. Notice that the first one, you can have a C or a T. At the second one, you can have a G or a T. And at the third one, a C or an A. And so we've got various different sequences in the population. And notice that on the top green chromosome, we have a beneficial mutation. That's the white A. The other ones we're going to say are just neutral variants. Now, I'm going to do this initial thought experiment with no recombination occurring, no meiotic crossing over between these chromosomes. What will happen? Well, it's actually super simple. Remember, selection is favoring the white A. Okay, selection favors the white A, which means we're going to basically eliminate everything which does not have a white A, and the individuals with the white A will take over the population. This is the fixation process. So that's natural selection eliminating all the other chromosomes. So at the end of the day, what happens? Everyone has a white A, that's natural selection, but it's not that everyone has the white A. Everyone has the same sequence on either side of the white A. And the term of art we use is the T at position 1, the G at position 2, and the C at position 3 have hitchhiked. They've come along for the ride with the white A. So the footprint, the genomic population genetic footprint of a adaptive event like this is a complete loss of genetic variation in the region. the whole, in this case, because there's no recombination, it's just one chromosome, the original chromosome where the white A arose that has gone to 100%, has gone to fixation in the population. But you say that's a little extreme because we know recombination occurs. So let's now go on and look at the same situation with some recombination, okay? And so what we're going to do is, again, exactly the same scenario. We've got the white favored A, and we've got a diversity of chromosomes in our starting population. Now, the difference, if you've got recombination, is notice now at the top, the green A-bearing chromosome is associated with a turquoise chromosome, and we can have a crossover event in meiosis. So let's say it occurs there. What happens? Now, notice we've got a chimeric chromosome. So we've switched the T and C between those chromosomes for position 1. Now, and obviously this can happen elsewhere. Let's try another chromosome. So here, this is a crossing over which switches the position 3 alleles, and we get something that looks like that. Now, notice recombination is a rare event, so it's much more likely to occur at some distance away from the A locus. In other words, you're more likely to get a recombination event between the advantageous locus and a distant one than between one which is very close, just because recombination events are rare. So notice that even though A is no longer just associated with T in position 1, it's also associated with C in position 1. And even though A is no longer associated with just a C in position 3, it's also associated now with an A in position 3. It's still only associated with a G in position 2, because position 2 is that much closer to A. Okay, so what happens? Well, same thing. We're going to have natural selection do its thing. If you've got a white A, you're in good shape. So what does that mean? Eventually, the entire population will come to look like this. Now, notice it's the same footprint. We've lost variation for the A mutation. Obviously, everyone's got an A mutation, but notice at position 2, everyone's got a G mutation. But the further you get away from A, so there's a bit of recombination. So at position 1, you can have a T or C, and at position 3, a C or an A. So this means, rather than the extreme case that I gave you earlier, where the footprint of positive selection was the entire chromosome was identical, what we have here is a chunk of the chromosome flanking the advantageous allele, the white A in our case, and that's going to taper off, if you like, as you get further and further away from the A locus. Okay, so that's entirely hand-wavy and theoretical. Let's see some real data. This is kind of cool. This is looking at the malaria parasite, one of humankind's great enemies. And as you know, we try and kill malaria with various drugs, various anti-malarials. And this is the bad news about the effectiveness of natural selection. Malaria evolves to cope, to be resistant to these medications. And this is a well-known case of a gene called DHFR, which is now known to be implicated in malarial resistance to an early drug. And what you can see, natural selection has operated on malaria population. So in the area immediately on either side of the DHFR gene, you've almost got zero variation. The vertical axis on this graph is the amount of population variation there is. And as you go further away from the DHFR gene, so you see an increase in variation. So this is the footprint of adaptive evolution in recombining DNA. Reduced variation at a population level, at a species-wide level, in areas flanking the adaptive trait. Okay, third and final way to disentangle, if you like, genetic drift from adaptive evolution, from positive selection in terms of fixation, driving a mutation to 100% in a population, is ancient DNA. This is the coolest technology, as you know. So we can recover usable genomic sequences from truly old material. The current estimate is, I think, probably about 500,000 years is about as old as it can get. And there are all sorts of issues with this. The big one is this, is that DNA essentially falls apart once you're dead, okay? It degrades, it breaks up. So traditionally, everything was thought to be too small, too shattered, too fragmentary to be able to do any analysis of this kind of material. But with high-tech next-gen sequencing, the problem is solved. And let me just show you sort of schematically why. Let's say here's a chunk of chromosome 5 from a bone of a Neanderthal. And so these are 4 different chromosomes 5. But over time, as I say, it's degraded. So we've only got little fragments of each one. What we can now do, and it's an amazing technology. This is brute force. We can sequence every one of those fragments. So this is a massive sequencing undertaking, one. And then the next piece of brute force is computational. We can align all those sequences, basically find all where the overlaps between each one occur, such that we can reconstruct entire genomes. I'm sure you know this. It's amazing. And we found out all sorts of important things, including, for example, that there's this relative of ours, which we know nothing about or knew even less about. This is the Denisova cave in Siberia, from which a pinky bone and a tooth were recovered. Turns out to be from a distinct relative of Neanderthals. So another close cousin of our own species. Here's the pre-Denisovan. We don't know what this person looked like even, right? Because we have that much of material and that much of material. So this is the pre-Denisovan phylogeny. I've gone for Beyonce now, upgrade on Miley Cyrus. There's Homo neanderthalensis. Now we've got a new member of the family tree, the Denisovan. Fine. Well, that's sort of, that's of interest to people who study fossils and human evolution. No, this is of interest to us all. Because, and again, you know this, because we have the Neanderthal genome and we have the Denisovan genome, we can go into your genome, we can go into my genome, we can go into everyone's genome and identify bits of our genomes that were derived from Neanderthals and Denisovans when our ancestors, modern humans, first emerged from Africa, maybe 80,000 years ago, met Neanderthals, met Denisovans, and had sex with them. Okay, so that's ultimately where the segments of your genome, which are derived from Neanderthals and Denisovans, came from. Okay, now why am I telling you this in the context of positive selection? Because, and this is super cool, we can use ancient DNA information to identify the sources of adaptation in our modern populations. This is a complicated slide. I apologize, but it's kind of cool. Tibetans. If you've ever spent any time at high altitude, maybe even when you're skiing on Uludağ or Acacias, high altitude is exhausting because there's very little oxygen. But Tibetans who live at high altitude, they live at 3,000, 4,000 meters, are fine. Now, why? It turns out it's largely to do with a adaptation at a gene called EPAS1. And so this is the trick that Tibetans, the genetic trick that Tibetans have. That's super cool. But the really cool thing about EPAS1 is the sequence that Tibetans have, the gene is very different from other humans, quite distinct. It turns out that it came from Denisovans. So as you can see on this tree, there was an interbreeding event with Denisovans with the ancestors of modern Tibetans. Tibetans got the Denisovan EPAS1 allele, which is adapted to high altitude. Now, this was a bit puzzling to start with because Denisovans, the cave in Siberia, is not at high altitude. So it's only recently, last year, actually, I think, that we found another piece of Denisovan material. And that is a jawbone. You can see that photograph there. And that jawbone was found in a Tibetan cave at, I think, 3,500 meters of altitude. So the Denisovans lived at altitude. They evolved to thrive at altitude and cope with that lack of oxygen. And then through interbreeding, they passed that adaptation on to the ancestors of modern Tibetans, who still thrive at that altitude, courtesy the Denisovan adaptation. Ancient DNA is the coolest stuff out there, and is one of several tools we can use in our attempt to understand and identify instances of positive adaptive evolution, positive selection. So that's all I have to say. It's going to be a slight change of speed now as we go to Naomi's talk. I will hang up that. Yes, just so you know, we're going to have questions at the very end of the session. So please save your questions until after Naomi's talk. Good. Thank you very much.
Thank you so much, Professor Berry, for your brilliant talk. Now we will move forward with Professor Pierce's presentation and talk, as you said. After that, we can have a short Q&A session. So, Professor Pierce, welcome again. If you are ready to, we can start now.
Okay, it's very nice to be here again. Thanks for having me. And it's fun to, it's hard to talk after Andrew because he gives a great talk. But I'm very happy to share with you some of the research that we've been doing in my lab. And I think I'll just go straight to this slide. So let me just select that here. And I'll go to the screen. There we go. Okay, so hopefully this means I can just go straight in. Whoops. Oh, I guess I'd better start at the beginning, huh? There we go. There we are. Okay. So now for something completely different. In my lab, we study species interactions, which of course are the fabric that ecosystems are formed from, different species interactions, but they're hard to study. And for many years, people have worked on things like ant plants because they're a little more tractable. At least in nature, you can study them, the ants and the plants together. But there are difficulties in working in nature, but I'll get into that in a minute. The work I'm going to talk about today took place in Kenya, and quite a few of my students were involved. So I talk about them first. Dino Martins, especially, was a former student who is Kenyan and introduced me really to this whole system, and drew all the others involved. And their names are up here. They all participated in the experiments that I'll talk about, and especially Richard Childers there. This is mostly from his thesis. Okay, so we study ant plants, and we study ant plants because they are classical examples of mutualism. You can see very easily that the plant offers something for the ant, and the ant takes care of the plant. And there are many different species. Andrew talked about convergence. Well, there are more than 680, probably more than that, examples of these myrmecophytic or ant plant species around the world. And quite a few of those contain ants that we call phytoecious. These are ants that are never found free living. They're only ever found living inside the plant. So they become obligate partners of the plant. And one of the really interesting things about ant plants is that they're restricted in their distribution to the tropics. And really, we do not know why yet. But let's go to East Africa. Here we are zooming in, where Vachellia drepanalobium, the African whistling thorn ant acacia, is the dominant tree found across all the black cotton soils. These are vertisol clay-like soils spread right across East Africa. As far as the eye can see, nothing but these trees. And if you look down when you're flying in, you see that they're over-dispersed. The trees tend not to touch each other when they're growing naturally. And this is because each tree is effectively separate ant colony. So the trees are dispersed evenly across literally thousands of kilometers of East Africa. But there are commonly at least 3 different phytoecious ant species that live in those trees, and they form a mosaic living in the trees throughout East Africa. You can see that it's immediately just a fascinating system for looking at the evolution of species cooperation and also coexistence, where these 3 different ant species, 2 chromatogaster species, Mimosae and Nigerceps, and 1 Tetrapona, the all-black one, are sharing a resource. Not something that's supposed to really happen in ecology, so immediately we had to start studying why. As I said, it's an example of a classic mutualism, even from the 1960s, from work by Jansen in Central America on ant plants. It's been shown, the ants benefit the plant by patrolling, grooming, and defending the plant against herbivores. And the plant provides these domatia. These are little houses. And in the case of the whistling thorn, they're quite big swollen stipules on the plant itself that the ants live inside. And they also give them food in the form of extrafloral nectar. And in some ant plants, they make little food bodies. So it's a basic sort of defense for food and shelter mutualism. Now, the system we study in Africa, we went there partly because this is by chance, partly because Dino convinced me to come and work there. But it was a very good system to work in because ecologists like Truman Young and Mo Stanton, Todd Palmer, Rob Pringle had done lots of work on the ecology of the system. So we came in with an interest mostly initially in genetics, and so lots had already been done. And one of the very first things that different ecologists had done was to see whether or not what did the ants, did they really protect the trees against herbivores? And of course, in Africa, this is a kind of special herbivory because many of the herbivores are not insects eating plants, but really these large grazing megavertebrates like a giraffe. And you may say, well, I can't believe the ants would make a difference against a giraffe. But here you can just see in this simple graph where Dino had followed around the giraffes that were grazing on the trees and found that the giraffes approach all the trees with the 3 different ant species pretty much the same, but they don't stay the same. If the tree has Mimosae in it, this is the red and black ant with the one with the red head, they don't stay and browse on it. They positively avoid that tree. And this is true also of rhinos and even elephants. And just to give you a sense for why that might happen, this is what it looks like if you come up close. They don't seem to be too bothered by the thorns, but if you get a nose full of these biting ants, it's really very, very unpleasant. So they really are effective against herbivores. Now I'm going to talk about 3 things, and Andrew's already slightly given away that we're talking about plant odors, but we came to this, to looking at this particular adaptation in a sort of circuitous path. So, and I just wanted to give some background about the system so you could understand the context in which maybe it was not such a surprising discovery. One of the things that interests us coming into this system was that it wasn't just a classic 2-species mutualism. There were a lot of other parties in this network. So maybe a network of interacting species that predominantly looked like a 2-species interaction, but not really just 2. So for example, these are 2 caterpillars of Lycaenid butterflies. These are, we also studied these because they have a very complex interaction with ants and are usually not attacked and eaten by ants. And after studying for a while, we found that there were at least 25 species of these Lycaenid butterflies that parasitize the ant plant mutualism by laying their eggs on the trees. And the caterpillars are not attacked by ants. The ants actually protect them against other enemies, and the butterflies do very well on the trees. When we saw that, we thought, well, it might be interesting to look at all the other things that are living inside these ant houses, sometimes right alongside the ants. So we decided to survey to look for what are called myrmecophiles, or ant-loving insects in the trees. And this was fun, really fun, because you can go out and collect the whole tree and bring it back to the lab and sort of take it apart slowly, as we're doing here. And this is what you come up with at the end, all these different myrmecophiles that are living inside those little ant houses. Now, when I first started as a biologist, faced with this kind of these data, it would have been hopeless, because these are, you'll see, they're all larval forms of different kinds of insects, except the spiders, of course. You know, no hope at all of figuring out what all of those things were. But of course, with the advent of sequencing and barcoding, it was just a simple matter of collecting a lot of trees and collecting a lot of myrmecophiles and then sequencing them, building a library of sequences against which we could compare to see what were the insects living inside. And we could come up with about, in one sample here, for example, about 82 different what we call OTUs, or operational taxonomic units, which are signatures of different species that live inside the plants. And you can do a little bit of estimation to see, you know, have you collected all the biodiversity that's there? And the answer is the curve is starting to flatten out, but not yet. No, there's really, these trees are occupied by many things that are attracted to the shelter of the ants, or some food that they get from the ants, or some byproduct. So they're host to many, many myrmecophiles, which in this case are mostly parasitic. You can't say that they're benefiting, they're parasitizing the mutualism itself. And actually, one of the things the striking thing that came out very clearly is that the different ants are parasitized often by very different things. And particularly this mimosae is hit by quite a few different kinds of insects and other sorts of parasites. So we have pretty specialized ones, and this is just a way of visualizing that. You can see from the different colored dots that these trees occupied by the 3 different ant species had very different inhabitants inside them. So they're specialized communities. Although we did also find generalists, and actually my favorite generalist is this ant mimic up on the top right. The one in that little image, you see an ant, that's Chromatogaster mimosae, and right beside it, a spider that mimics the ant so well that it took us a moment. I mean, when we see it ourselves, we can hardly recognize it. We thought, of course, this one must specialize on mimosae, because it has the red head and the black gaster, the abdomen of the ant. But actually, it was a generalist. It was on all of the trees with all the different ant species. And then you think, well, of course, it's trying to fool a predator. It doesn't matter so much whether it looks like the ant itself. It's not trying to fool the ants necessarily. So you learn lots of things from doing this kind of survey. What we also found was that it wasn't just insects that were in these trees, but in fact, a whole cocktail of microbes. And by microbes, I mean, I start first with fungi, because these interested us from the beginning. We just took the domation, we streaked out, you rub out the inside and just streak it onto a plate and grow what's there. And we did this because Dino, when he first started this work, had noticed that when we grow the ant colonies in separate nest boxes in the lab, the colonies were often lined by a white fungus that was of intense interest to one of the species, this Tetraponera, the all-black species. And that interested him because you can see they're tearing it apart here. It interested him because that species is a bit odd. It had never been, its diet had never really been established. It's hardly ever seen feeding on herbivores. It doesn't patrol. In fact, it's not a very good mutualist at all, as far as we can tell, in terms of protecting the trees. And yet it's frequently there. It colonizes very well. What's it doing? Well, Dino thought that it was feeding on fungus inside the nest. And indeed, if you set up an experiment, as Chris Baker, another graduate student, did here, and you look at you offer up fungus to the different ant species, you give them all a test to see what will they do with different strains of fungi, and the ones that we'd cultured out. Well, it's the tetrapona that go and harvest them and take them back to the nest and feed on them. So I think that the answer here is yes. It's not quite like like the great big ant gardens that you see in the New World, but these ants are definitely getting some nutrition from the fungi. Now, are the fungi that unique in the different ant species? Well, we looked at this question by looking at trees occupied by the ants at two different sites. This Kitengela is one site, it's near Nairobi, and Mpala is another site. It's about 200 kilometers north of Nairobi. And so these are very distant geographically, but the trees are there occupied by the three ant species. So we sampled from each of the ant species, and you can see that the ants are doing slightly different things. Inside the Damascia, one of them has these little leaflets, and the other has something that looks like a carton nest, and the other one looks like frass. Okay, so we sampled from inside. And we also sampled, we had them growing in the greenhouse, so we also sampled them from the greenhouse here in Cambridge. Of course, now it's much easier matter than to look at the diversity of fungi growing inside the Damascia. And here, I'm portraying it just at the level of class. And as you can see, the community of the fungi varies very significantly between the different ant species. And it didn't matter that they were growing 200 kilometers apart from each other, that what mattered was what was the identity of the ant living inside the nest. And I think this animation sort of captures this idea a little bit better. On the right, we have the fungi from the different ant species, and then the green ones are the ones that were on the leaves. They were rather more distant, but rather specific, or not. Let's see, the purple ones are the ones on the leaves. And on the left, of course, we also sequenced and looked for bacteria. And indeed, they also had rather specific bacteria associated with the ants. So there's a complicated garden growing inside those little ant nests, and exactly what the function is of all those different partners, we're not really too sure. You can immediately imagine, well, you have very different substrate inside those Damascia, inside the little nests. So maybe that's why there's such a different cocktail of microbes growing there. But what are they doing? And how did they get there? To look at this, we thought about, well, how did the nests form in the first place? Well, foundress queens of the different species of ants fly and find the Damascia. They find them when they're young, and they drill in. They make a hole and drill into the Damascia. So if you take a young Damascia and you look at the inside, at what's growing inside, they're pristine. We found no fungi or no bacteria inside the young Damascia before the ants had drilled in. But once they drilled in and had lived there for a little while, then we could find the gardens. It seemed to me that the ants might be carrying their fungal gardens into the Damascia with them. And to look at this, we looked at a particular gland or pocket in the ant called the infrabuccal pocket. And this is just a cross section made by Johan Billen, who's a brilliant does brilliant histology, to look inside that pocket of the infrabuccal pocket that is right. It's right beyond the opening to the mouth of the ant. And in many species, you can find interesting things in the pocket. What I knew was that in the case of Atta, the fungus-growing ant in South America, that when a queen founds a new colony, she carries the fungal hyphae with her, and she inoculates the new colony herself. And sure enough, if you see these, the different, the materials here inside, they look like hyphae, and when we sequence them, at least here for Tetraponera, and here for Chromatogaster nigroceps, so for 2 of the ant species, we could recapture all the diversity that we found inside the little houses simply by sequencing the infrabuccal pockets from the founding ant queens. Now, why is this interesting? Well, of course, we've all been brought up as ecologists to believe that ant plants are a beautiful example of a defense mutualism where the ants take care of the plant and the plant houses the ants. But what is a true mutualism in this system is actually between the plant and the fungi. I mean, we know very well from other kinds of fungal interactions that fungi can be beneficial for plants. And so if the true mutualism is between the fungal garden and the tree, then maybe the ants are just the vector that carries the fungal garden to the tree. Now, to look at this, here you start to see the complexity of studying something, even the simplest, what seemed like the simplest 2-species mutualism suddenly gets very complex. So we grew up all the trees here with the plan to now inoculate them with fungus in the bush house here and not let them be, so we did the inoculating instead of the ants. And we were all ready to have this experiment run. And this was just as the whole world was shut down by COVID. And here's Ivy, the person we worked with there, sending us a picture of how beautiful the plants were, but unfortunately we were unable to work with them. Okay, so this is a diversion that I mentioned at the beginning. We had this extra detour, what to do because we couldn't come over and we were stuck at home. Well, fortunately, we had carried back colonies of ants, and we could grow the plants in the greenhouse at Harvard from seed. So we could take the plants, the trees, as young trees, and we could inoculate them with ant colonies and just look at some of the effects that the ants might have on the plants. And so we did this, and very interestingly, and maybe not surprisingly, because we had a hint that this might happen, we found that yes, the different ant species have a very strong effect on the architecture of the plant, on the way it grows, both in terms of the size and the shape of the domatia. You can see that on the left-hand graph, but also even in the number of domatia formed, as you can see on the right-hand graph. So, and we weren't too surprised by this. In fact, it had been written about in the literature quite a bit because people notice driving around in the field that when the tree is occupied by one ant species or another, they often take on a very different appearance. And this is just showing that, yes, somehow these ants are influencing that, that growth trajectory. But this led to thinking about how it is that in a good mutualism, the 2 partners are often very sensitive to the signals from the other partner. And I started to wonder whether the plants might also respond to the ants, simply to the smell of the ants, not the patter of little feet, but the smell of the ants. And this led to this experiment that Andrew talked about, which we've called the smelly ant experiment. And I have to preface this by saying that this is one of the hardest experiments in an odd way that we've done. And we did it 4 times just to make sure that because we almost couldn't believe it ourselves. As you can see on the left here, we had ant colonies in little artificial nest boxes with a screen mesh top. So it was possible to put an ant colony inside the plastic bag here with a growing seedling without having them touch each other. So the ants never crawl on the plant. The plant's just grown from seed, so it's never experienced ants on it before. But it's grown in the atmosphere of the ant. And we would do this, we would put it in a bag every morning for an hour and a half until the bag started to become moist with the humidity and so on. And then we took it out. So for 6 weeks, just an hour and a half every morning, the plants were exposed to the smell of a different ant, each one with its own ant species. Now, as I said, we did this experiment 4 times, and that was because we kept thinking of things. So, well, other possible explanations. For example, we also controlled for CO2. We took that into account anyway, because we thought, well, the colonies are slightly different sizes, so maybe there'll be a difference in respiration rates, and we weren't thinking about that. But no significant difference, and we did do this very carefully. The reason we were careful was because we got a very significant difference. And that is that when we grew the plants with their symbionts, they branched significantly more often. So with the symbiont, you can see it. If you look at the example of the plant on the top panel here, it has many branches, whereas the plant at the bottom panel doesn't. It was stunning. You could see it even in the greenhouse as they were growing. And the reason that branching is nice, it's easy to count, of course, and you don't have to worry about measuring length, although we did that too. But the branching shows you that what it's doing is it's amplifying the number of growing tips on the plant. And since both the extrafloral nectaries and the domatia are formed by growing plant tissue, this ensures that extra branching ensures that the tree is going to make more domatia as it grows. And it will also make more extrafloral nectaries, except in the case, and on the right, we found for one of the ant species only, this Tetrapona apensigi, it made fewer extrafloral nectaries than in all the others. This was also surprising to us because Tetrapona apensigi is the one species that does not feed on extrafloral nectar. So the fact that the plant doesn't reward it seems amazing, amazingly fine-tuned adaptation to the smell of its partner. Of course, this raises lots of questions about the plants themselves. How could they perceive the ants? What are the chemicals that they're responding to? And especially, what is the nose? Where's the receptor on the plant to these chemicals? I'm not so surprised. I have to say that, the signaling pathways are highly conserved across the tree of life. So the fact that there are signals that can transmit here between the ants and the plants are not so surprising. And when we looked at the composition of the headspace or the volatiles coming off the plant, there were something like 275 compounds, but a few of them are very outstanding ones that are certainly candidates to look at more closely for this to understand this response. But still, and what the receptor? These are really still some outstanding questions that we don't know about the natural world, and it's been very fun to do this work. I'll just summarize quickly to say that I hope I've convinced you that both partners in this system are co-evolved and are very responsive to each other. Also that there are many third-party associations in these classic mutualisms that make it difficult to assign costs and benefits here. I mean, for example, mimosae may protect the plant better than all the others. It's very ferocious, but it also harbors many, many myrmecophiles. And if it has many herbivorous things living inside it, well, is it really better for the plant? It's a complicated set of costs and balances. So the outcome of any of these interactions is quite context dependent. I also, I think maybe the most novel aspect of the work that we've shown here is that the plants can respond to the smell of their symbiotic ant partners. But really, we need to do a lot more work to understand how they are able to do that and and what the plants are producing. And also how widespread is this ability across plants in general. So thank you very much. These are, as always, the people who've done most of the work. Our lab group picture has turned into the, you know, the typical Zoom shots, and here we all are. I also had lots of help from family. You can see Andrew and our daughter Katie behind me. Were out there to do this work, and I'll stop there. Thanks very much. I think I can
Thank you very much for your presentations. Not surprisingly, they were very enlightening again. And during your presentation, our team compiled the questions from the chats. We would like to forward some of them to you if it's also suitable for you too.
Great.
Okay, so Tuğçe or Drew.
Yeah,
you can start.
First, I'd like to thank you very much about the presentations. Both of them were very informative, and I really enjoyed watching them. My first questions for Professor Berry. How should we evaluate neutral mutation to detect selection at the molecular level?
Sorry, say again. How should we evaluate mutations effect
neutral
at the molecular
mutation
level?
To detect selection at the molecular level.
How mutation affects selection at the molecular level? Well, it's somewhat predictable if, you take a piece of DNA and you do some functional genomics and proteomics such you know what the different segments of the piece of DNA are for. So you've got exons and you've got introns and you've got upstream and downstream regions and so on and so forth. So you can predict to some extent the impact of a mutation upon that piece of DNA. If it changes the encoded amino acid, then probably that's going to be deleterious. As I said, you can't mess with something that's working unless, again, maybe you can change an amino acid and it's the same sort of functional, same, say, hydrophobicity or whatever, so it doesn't have a big impact. But typically, for example, changes in third positions, redundant positions, courtesy the genetic code, are again probably neutral. But this is the amazing thing about evolutionary genetics. You might say, yes, it's in a third position, it's completely redundant, it doesn't matter whether you have an A, T, G, or C. But we now know that in many instances, for many genes in many species, we have what's called codon bias. Codon bias is where it matters. So let's say you've got a serine codon, and you can have either A, T, C, or G. It's all serine, but it turns out it's better to have the C in that third position. Now, that seems crazy because you've still got a serine, but codon bias tells us that there's selection in favor of having a C, it's still a serine, over an A, T, or G. And that there are various theories about why that is. It's probably related to translational efficiency or whatever. But so obviously I could go on. This is the answer to the question would be a textbook. But I just want to say that to a certain extent, we can predict the impact of a mutation given where it lands in the genome. But to a certain extent, we cannot. So as I say, if it's in a third position, you might think, oh, fine, it's a neutral mutation. But if there's codon bias, it absolutely is not. So the short answer to the question, Duru, is an ongoing process of exploration. The more we understand about the genome, the more we understand about selective constraints on genetic variation, the better picture we get on the impact of this particular mutation on this particular piece of DNA.
Okay, thank you very much.
To Professor Berry, we have another question. In the light of the information that you have given on lactose intolerance and the effect of domestication on it, can we say that in populations that have civilized early and quickly, their increased ability to adapt to modern life is somewhat related to this kind of mutation and positive selection?
No. The interesting thing about lactase is it arose first, or lactase persistence, I should say, it arose first in populations where that was needed, where cattle or goats or camels or whatever had been domesticated, where there was a permanent supply of lactose. So this is how evolution works. It's serendipitous, right? It's just by chance. Yes, people in, close to Anatolia had domesticated cattle. So then there's a need for those people. There's now selection in favor of lactase persistence. Now, whereas people in South America had not domesticated cattle, they didn't have an endless supply of lactose, so they didn't need lactase persistence. Now, that doesn't mean to say that those people are in some way less civilized or whatever. They're doing other things. Remember, for instance, the Central Americans domesticated maize, corn. So you've got different societies which are doing different things culturally. Innovation occasionally spreads, yes, through communication and so on, and also migration. But the idea that one group is more capable of responding and evolving more readily to cultural changes, to innovations, to new ideas, there's no evidence for that whatsoever. What really we're talking about here is, in fact, not biological evolution. Biological evolution is what's occurring in lactase. There's a mutation, a specific mutation, which changes the switching of the lactase gene, and it's advantageous to keep it switched on. Fine, that's easy. But it's incredibly slow and inefficient biological evolution. Think about it. So let's say I'm the first person with a lactase gene switch that keeps it switched on. So, and times are hard. We need milk. My kids survive. Your kids don't, because we've got milk, but your kids can't process it. Okay. So what does that mean? I'm going to have more grandchildren, and then more great-grandchildren, and so on. But it's generation after generation after generation after generation will slowly see the increase of the favored variant. Okay. Now compare that to cultural evolution. Let's say I invent the wheel. Okay. I don't need a mutation to adopt the wheel, right? I see the wheel. You look across at me and say, that's a good idea that Berry's had, inventing the wheel. So you copy the wheel, and then everyone copies the wheel. So in literally a short period of time, these innovations, these, you might say, attributes of civilization spread across populations. It's nothing to do with biology. It's everything to do with our super smart brains. We learn stuff, we acquire ideas, we then, if you like, implement those ideas ourselves. So I think it's important to distinguish here between biological evolution, which is slow and serendipitous and clunky, frankly, in the context of the very short time frame of human evolution, and human cultural and otherwise innovation, and that is cultural evolution, and that is virtually instantaneous, especially today. I mean, think about it. All you need is your favorite influencer, I don't know whoever it is, who says you should do your hair like Dr. Berry, and everyone wants to have white hair and a white beard or something like that. And it's sort of instant response of adaptation. I mean, not that that's adaptation, but cultural change in response to a cultural leader.
Okay, thank you very much.
Now I have another question for Dr. Pierce. Can the ant types go through a behavioral adaptation to replace niches, such as in a situation like the absence of type X ants, but the abundance of type X trees? Will type Y ants will act like type X ants to continue the mutualistic relationship and take over the biological niche?
Well, that's a good question. I mean, in the case of the 3 ant species that we were studying, and there is a 4th that is sometimes in the habitat, they are all symbiotic and in a fixed way. They can't live anywhere else except in the trees, and they do benefit, or they benefit the trees enough that the system is maintained right across East Africa. But there's an invasive ant species that has been wiping out these co-evolved species that has taken off within the last 5 to 10 years. That is, and this is Phydolemaccephala. It's a fairly common, widespread pest ant that it's something of a puzzle because it had been recorded to be present in Africa for a very long time, but something triggered these colonies to go into an invasive mode, and they are killing the native ants in the ant plants, and they do not provide services for the plant. And there's no sign that that is evolving or taking place. So the only good news there is that it does seem to be happening very rapidly, and because the ants reproduce very quickly, it's not good now for the ants are sweeping through and trees are dying, but the ants move on to find the invasive ants move on to find more food, and if they leave devastation in their wake, but the trees then later come back, and ants are present. So it's a dynamic process, but clearly not such an easy thing to become an ant plant ant. It takes a little. We're sort of seeing that evolution in action, and I'll be interested to, if you ask me this question in another 10 years, be very interesting to what's happened in this system.
Okay, thank you very much for your answer.
Okay, now another question to Professor Pierce. Does the mechanism based on the smell of plants occur only between plants and animals, or can there be an interaction between different plant species or even the same species?
Yes, definitely between different plants, and that has been known for a while. There was a famous science magazine cover of showing a stand of a clone of trees. It was called, and the title was "Talking Trees." So this was a study done by Ian Baldwin, where I think he was a graduate student then, where he noticed that when gypsy moth attacked a set of trees, that the neighboring trees would become more resistant to the gypsy moth, and he thought that must be because somehow the attacked trees were releasing volatiles that the trees that had not yet been attacked, these were of the same species, it would cause them to mount a higher immune response, a sort of cascade of signaling responses to defend themselves against the gypsy moths. And there's been lots of work done since then to show that in fact can certainly happen between different plant within a plant species, and also even between. There's a famous study of a greenhouse study of tomato plants signaling to sagebrush plants. I mean, as soon as the tomatoes were attacked, the sagebrush plants in the same greenhouse started to upregulate their defense compounds and so on. So that is, I would say, definitely known, and it does seem to be quite widespread. And one of the main volatile chemicals involved is called methyl jasmonate, and it's part of the jasmonate signaling pathway that will trigger the production of defense compounds in a lot of different plants. But what is less well known, and I think maybe there are only a few examples that I can give you, is this idea that the plants are responding to insects, or to so it's really across very different realms of life here, and that's a little less common. There's I think the other example I can think of is of a gall fly that lives on goldenrod, and the goldenrod will respond to the smell of the flies. But to my knowledge, that's the main other example I can think of. But people maybe haven't looked, so there's a lot to do here.
Thank you very much.
Because of the time limit, we have to stop here, unfortunately. But it was very special for us to see you again and have you here as our first talk of the series. Thank you again so much for your collaboration and your contribution.
Thank you all, and good luck with the rest of the series. It's wonderful work you guys are doing. Thank you.
Yes, yes,
Thank
good luck to you. Thank you. It was a pleasure to be here.
Yayınımız saat 8'de Marta Kluki'nin antibiyotikler üzerine olan konuşmasıyla devam edecek. 8'de görüşmek üzere. Bye. Thank you again. See you.